14,000 years of isolation forged a genome that exists in no other population on Earth. Then one migration rewrote 80% of it. That ancient DNA should have vanished. Instead, it survived where no one expected. At the end of the last ice age, rising seas cut the Japanese archipelago off from the continent. The land became a chain of islands, stretching from the snowy forests of Hokkaido to the subtropical shores of Okinawa. Archaeologists have traced the earliest foragers to coastal sites like Minato-gawa Cave in Okinawa, where stone tools and layers of ancient shells date back nearly 13,500 years. These early inhabitants built their lives around the rhythms of the sea, leaving behind shell mounds and middens that still mark the shoreline. Over thousands of years, their settlements grew more complex, with pottery fragments and traded obsidian revealing networks that spanned hundreds of kilometers. For almost 14,000 years, these islanders adapted to shifting coastlines and changing forests, their communities persisting in relative isolation. The uninterrupted human presence, anchored in the evidence of coastal camps and middens, set the stage for a genetic legacy found nowhere else on Earth. Genetic analysis of ancient remains reveals signatures that set the Jomon apart from any other population in East Asia. Among Jomon males, between 40 and 80% carried the Y chromosome haplogroup D1b2, a lineage almost absent outside the Japanese archipelago. High-coverage sequencing from petrous bones confirms this frequency, with a best estimate around 60%, varying by region. On the maternal side, mitochondrial DNA points to N9b as a defining marker. This lineage, present in about 12% of modern Japanese and even higher in ancient samples, traces directly back to the earliest settlers. Stable isotope ratios from Jomon bones show a diet with more than 70% marine protein, underscoring a lifestyle deeply tied to the sea. These genetic and dietary markers remain embedded in the DNA of modern Ainu and Ryukyuan people, making the Jomon legacy an irreplaceable foundation in the genetic landscape of Japan. Around 2,900 years ago, the islands of Japan began to change. Farmers from the Asian continent arrived, bringing rice seeds, new tools, and unfamiliar ways of life. Archaeologists trace their entry along two main corridors, one sweeping up from the Korean peninsula into northern Kyushu, the other threading through the southern islands from the Yangtze Basin. Radiocarbon dates from the Itazuki site in Fukuoka show rice paddies in use by 2,900 years before present, cut into the earth beside stone tools that differ sharply from those of the Jomon. Bronze mirrors, analyzed for their lead isotopes, point back to southern China rather than Korea, hinting at long-distance trade routes that crossed open water. Nakagomi's map of migration routes shows both the northern and southern streams converging in Kyushu, then fanning out across the archipelago. The physical traces, paddy fields, imported artifacts, and the spread of new pottery styles make clear that these were not isolated contacts, but the first waves of a migration that would reshape the genetic landscape of Japan. Genome-wide studies of modern Japanese reveal a striking signature left by the Yayoi farmers. The Y chromosome haplogroup O2b, a marker common among ancient rice farming populations from the Korean peninsula, now appears in about 45% of Japanese males. On the maternal side, mitochondrial DNA group C, also linked to continental East Asia, dominates the mainland. When researchers model the ancestry of today's Japanese using QPADM and similar methods, the numbers consistently point to a major continental input. Roughly 80% of the modern genome traces back to these Yayoi migrants. The remaining 20% carries the imprint of the Jomon, but this fraction varies by region. These findings are not just statistical abstractions. They show up in the DNA of people living in Tokyo, Osaka, and across Honshu, where the farmer ancestry is strongest. As Professor Nakagome puts it, the genome is a mirror. It reflects both the seaborne hunter and the rice field farmer. This dual origin model, grounded in hard genetic evidence, sets the baseline for understanding why Japanese DNA stands apart even among its East Asian neighbors. In the far north and deep south of Japan, the genetic legacy of the Jomon endures most strongly. Among the Ainu of Hokkaido, recent genome-wide studies led by Masaharu Hanihara show that up to 55% of ancestry traces back to these ancient hunter-gatherers. In the Ryukyu Islands, Satoshi Kondo's research reveals a Jomon proportion around 35%, far above the mainland average. The Y chromosome haplogroup O1b2, a marker introduced by early migrants, appears in about 18% of Ryukyuans, but Jomon lineages remain prominent. These outlier populations challenge the idea of a single, uniform Japanese genome. As Hanihara notes, the Ainu carry the voice of the ancient people. While a Ryukyuan fisherwoman puts it simply, our genes never went inland. The genetic map of Japan is a mosaic, not a monolith. High-coverage sequencing of Yayoi period genomes, published in 2025, has pushed the story far beyond a simple two-way split. Researchers now detect 3 separate pulses of continental ancestry entering Japan. The first around 2,800 years ago, the second at 1,800 years ago with the main Yayoi migration, and a third about 400 years ago. Using QP graph models and direct radiocarbon anchors, the latest data reveal a split of roughly 60% Jomon, 20% Yayoi, 10% later East Asian, and 10% unique Ainu Ryukyuan ancestry. Petrous bone sampling yields over 30% endogenous DNA, allowing this fine resolution. As Chris Tyler Smith sums it up, Japan's genetic history is a palimpsest, layer upon layer, each migration leaving its trace. Japan's genome still holds secrets no other population can claim. As new DNA discoveries emerge, our understanding of identity and of human origins keeps shifting beneath our feet.